In Lab 21, we are covering blood vessel histology. So we have to think about the scale of things when we think about histology. In the previous videos, we talked about veins and arteries. Those things are visible from our muscle man model. But now we're getting more into the microscopic side of things. There's a lot more to see. So first off, as a reminder, arteries are going to be carrying blood away from the heart. Typically, it will be oxygenated, unless we're talking about that pulmonary circuit. And so you may also see this color coding on our our blood vessel man, that is. Similarly, veins are going to be taking blood toward the heart. So this is how blood is draining back to our heart through this entire system. So far, we've only seen these terms veins and arteries, but there are a couple smaller vessels to talk about. So our largest scale is the artery. Anything from our aorta all the way down to the arteries of the extremities, that all falls under this category. However, we also have structures called arterioles. So a lot of times we'll have our arteries with many, many branches coming off. As they keep branching, we start to call them arterioles, just little arteries. And then eventually those arterioles will split off into our capillaries, which is kind of the smallest scale for blood exchange. Capillaries are really helpful because they are so thin, they're going to allow a site of exchange between blood, specifically the red blood cells, and then the cells of our body. So the level of the capillary is where we actually have gas exchange, nutrient exchange, waste exchange. All of that happens at the level of the capillary. And then we have our venous system. So again, the largest scale that we've seen is the vein. Our superior and inferior vena cava are technically veins, they're just really big. And then as we branch off and get smaller and smaller, we start to call them venules, little veins. And then again, we have capillaries. So capillaries are associated with both our venous system and our arterial system. One additional thing to know about veins is they do contain valves. So it's really important that our blood is getting back to the heart, and it has to work against gravity a lot of time to do that. So we have veins built in, or valves built into our veins rather, that are going to prevent backflow of blood. So that's one unique thing about veins that we don't see in our arteries. We also have 3 common layers. So in any of the veins or arteries that we talk about, we can't expect to see the same 3 layers. How they look and how thick they are is going to vary between arteries and veins. So this diagram shows it off really well. This is nice and color coded for us, but we're going to essentially see the same pattern under a microscope today. The outermost vessel, whether we're talking about vein or artery, is always going to be our tunica externa. So we see that term tunic coming up again. We saw it with, for example, layers of the eye, and now it's coming back for blood vessels. So tunica externa, sometimes called tunica adventitia, is going to be this outermost layer. And it is made of connective tissue, so it'll look a little more fibrous under a microscope, and we won't have as many cells in this layer. So we'll look at that in just a bit. Every artery and every vein is going to have a middle layer called a tunica media. So tunic meaning layer, media meaning middle. That one's going to be a little easier to remember. And that is color-coded purple on this model here. The tunica media is made of smooth muscle, so what we would expect to see is this nice wavy pattern with muscle cells interspersed throughout. So the dark purple dots that you see here are actually the nuclei of our smooth muscle cells. So let's start to think about those associations, what we'd expect to see in connective tissue, which is acellular for the most part, versus acellular muscle tissue. So that is our middle layer. It does tend to be thicker in arteries. And the reason for that is arteries control blood pressure. And in order to change the pressure flowing through a tube, we need to be able to constrict the tube, make that area smaller, or dilate the tube and make it larger. So arteries, and specifically the tunica media here, the smooth muscle layer, they are responsible for constriction and dilation of the entire artery blood vessel. And that is what helps us to control our blood pressure. That is all tunica media, and then we have one more innermost layer, and that is called the tunica interna, or internal layer. Sometimes you'll see it referred to as tunica intima, so we can think of intimate as deep. If you're getting intimate with someone, you're getting to know them on a deep level. So that's how I remember this. Tunica intima or tunica interna is that internal layer. This one, because it is lining and opening, we still have a lumen when we're talking about blood vessels, which are just a different type of tube. Because it's lining a lumen, it is going to have endothelium, which is a very similar concept to epithelium. So epithelium, if you recall, is going to line a lumen or an opening, some sort of exposure to the environment. Same deal with blood vessels, only we have this different prefix now. Endo means within, so any of the epithelium lining a blood vessel lumen is going to be called endothelium. And so to summarize, we have these three layers in all of our blood vessels, arteries and veins. How thick they are is going to depend on which type of blood vessel we're talking about. So let's go ahead and look at some examples. There are lots and lots of examples in your modules, so I recommend looking at those photos, but we will also be using the Yale Histology site to look at these today. So we're going to start with arteries, and this is the biggest artery in the body. This is actually a section of our aorta. And so all of the stuff we would see here is typical of an elastic artery. So we're going to see this term conducting or elastic artery. This refers to arteries that are closer to the heart. So the aorta definitely counts as an elastic artery. And what makes these guys special is that they have lots of elastic tissue within that middle layer, that tunica media. So tunica intima, as we know, is always going to be that inner layer lining our lumen, which is in this case this big white space here. So we've basically cut our aorta in half, and that's what we're looking at here. So that makes this our tunica interna. And then we have just this really subtle wavy line. This kind of looks like a basement membrane, if you recall that term, where we're separating our epithelium from some underlying tissue, usually connective tissue. But in this case, we're separating our tunica interna from that tunica media. And so we're going to have a really big tunica media layer here, because this is where we would find our elastic tissue. And the reason this tunica media is so thick is that the aorta is dealing with a very high flow of blood. This elastic tissue is going to help the aorta to withstand all that blood pressure, because that's like the first point of contact with blood that's flowing out of the heart. And that's why this layer is so thick. And then we have our outermost tunica adventitia, which is going to be more of that swirly connective tissue layer. So if this is confusing right now, that's okay. We're going to look at some nice and labeled examples for us. Just wanted to give you the big picture first. So let's zoom in on that aorta wall, and we have to reorient ourselves here. So this is our aorta with that really thick vessel wall. On this slide, our lumen is right here, so it's on the right side. This is that innermost opening of our blood vessel. So immediately next to the lumen, we have our tunica intima. It's just a pretty thin layer of that epithelium and connective tissue. What we're really focused on when we talk about these elastic arteries is the presence of elastic connective tissue. So we can't zoom in super well here, but what we're going to see is a lot of these really squiggly lines. If you recall, elastic connective tissue is very squiggly because it has a lot of tensile and recoil potential. So that's what we would find in our tunica media, which is the thickest layer here. And then, continuing with that pattern, we still have our outermost tunica adventitia, or tunica externa. The same three layers, they just look a little different in the aorta. And now if we look at one of our examples from our module, the thing I want to focus on here is this tunica media. Again, we have the entire aorta, nice and thick, and then the thickest layer here is our tunica media. And in this slide, it does a good job of showing those dark elastic fibers. So this is what I want you to look at under a microscope when we're in lab, is focus on finding these elastic fibers. If you see them in an artery, that's how we know we're dealing with an elastic or conducting artery. So we're going to compare that concept now to muscular arteries. These guys are responsible for a lot of that vasodilation and vasoconstriction. So if we think about the term muscular artery, they're going to have a pretty beefy tunica media, that muscular layer. And this allows them, when that layer constricts, to either increase blood pressure when we make the tube smaller, or if this layer is going to dilate the blood vessel, that will make it larger and that'll decrease blood pressure. So the main concept here is that muscular arteries are responsible for that regulation of blood pressure. And what we have here is a cross section. Same 3 layers here. We're going to have a pretty beefy, smooth muscle layer. So their tunica media here is well defined. It's a little darker than the other layers around it, and this is going to be mostly smooth muscle. And then lastly, we have that outermost tunica adventitia or tunica externa. So again, same 3 layers. They look a little different depending on where we are in that blood vessel system. Last thing to note here that I haven't pointed out yet is typically there will be lots and lots of red blood cells in the lumen of our blood vessel. So it won't always be that nice empty space that we saw on this slide here. A lot of times our images will look like this, where we have lots and lots of those red blood cells. You'll notice that they don't have nuclei. So that's one way to remind yourself that we're dealing with blood vessels. They're going to have red blood cells in them. Don't let that throw you off when you're trying to identify arteries versus veins. Here is a zoomed in version of that muscular artery. So we're looking more at that arterial wall. We still have our lumen here. A little squiggly border of that tunica intima, which is a pretty thin layer. The thickest layer will be the smooth muscle layer. And note the lack of elastic tissue. Very different from that aorta we just looked at where we had a ton of elastic tissue. Muscular arteries don't really have elastic tissue. It's really minimal. We're going to have more smooth muscle than anything else to help with that regulation of blood pressure. And then lastly, we have our tunica adventitia. Okay. Let's look briefly at some capillaries. So this is not really testable, but I want you to kind of understand how closely capillaries interact with the rest of the tissue in our body. So what we have here is a capillary. It's really tiny, so it's really just a single layer of cells. And because capillaries are so small, they're only going to allow red blood cells to travel in single file, 1 cell at a time. So at this layer, because they are so small, they can exchange material really well with the surrounding tissue. In this case, we have lots and lots of adipocytes, those fat cells, in white. And then this is our capillary. So it's really thin. You can barely see the walls of it. And really what we are seeing here is these stacks of red blood cells filing one at a time. Because capillaries have such thin walls, they're going to allow really easy exchange of gases and liquids. So all of the stuff that has to happen at a cellular level to get oxygen from our blood to the surrounding body tissue is going to happen at the level of the capillary. So while this is not testable, we're not going to see this under a microscope, try to keep this concept in mind because we're going to talk about it with future labs. Let's switch gears now and we're going to look at some histology of veins. So I want to start at the largest level of the vein, which is going to be our vena cava. So it doesn't matter which one we're looking at right now, but this is going to be the largest vein in our body. We're going to see the exact same 3 layers within the vena cava and all of the veins that we look at. We have that innermost tunica intima or tunica interna. We will have our tunica media containing smooth muscle cell. We're really not going to see elastic tissue here because veins don't have to constrict and dilate. They're just getting blood back to the heart. And then we will also have this outermost layer of tunica adventitia. If this is confusing to you, how we go from one to the next, I want you to look at the number of cells we can see in each layer and the texture. So the tunica media here has these nice layers with smooth muscle cells. The dark purple here is those nuclei. That means this is a mostly cellular layer. This is going to be that tunica media. Tunica adventitia is made of connective tissue, so we're dealing with more of this kind of loosely woven, wavy, largely acellular tissue. Look for that pattern, look for the presence of cells versus the lack of cells, and then the texture of the tissue to help you separate those layers. So this is typical of what we will see with a lot of our veins. We have a couple differences from arteries to look at. What we have on this slide is an artery versus a vein. And the first thing I want to point out is the shape of these vessels. Arteries hold their shape well because they have a lot of elastic tissue. So they're going to hold that round shape really well. Veins are a little floppier. So the vein you see here is a little deformed. It doesn't have that robust elastic tissue in the tunica media, and usually that tunica media is just thinner in general. So it's not going to hold its shape well. Sometimes we will see valves as well within the vein. But the main thing you want to look for is the shape of the lumen, whether it's holding that round shape or if it's a little floppier. And then you also want to look at just the thickness of the vessel. Veins, as you can see, are going to be much thinner. So let's keep going. Another comparison we can see here, this is between an artery and a venule. Artery, again, is going to be pretty round. What we have here is a decently thick, smooth muscle layer, that tunica media, whereas the vein is much floppier. You can see it's not very round. And then the smooth muscle layer is really only 1 or 2 cells deep here. So just this layer we see surrounding the opening, that's going to be that very tiny tunica media. So let's briefly summarize what you need to know in terms of microscope questions. So we spent a lot of time looking at conducting versus muscular arteries. I want you to understand the differences there, where we have the presence of elastic tissue in one, we have a more robust tunica media with smooth muscle in the other. Also think about their location in the body, where we would find elastic arteries that are resisting high blood pressure versus where we would find more muscular arteries that are monitoring peripheral blood pressure. We contrasted the shape of arteries and veins, so your module will be very helpful for this. I want you to be able to look at a slide like this and identify the vein versus the artery and explain to me how you figure that out. So are we looking at things like the shape of the lumen? Are you looking at the thickness of the tunica media to help you identify those? If you're looking for additional practice, Yale has a really nice virtual microscope. So this goes a little more in depth than what we need, but I want you to be able to look at a cross section like this and note whether you're looking at artery, vein, and how to tell those apart. So I hope this helps, and good luck reviewing.